Hello and Assalamualaikum to the examiner. I am Nur Syakirah Syahrain from FYP2. In this video, I'm going to present my final year project, which was a study of stability of Kenaf leaves herbal tea integrated with Prunus Persica or peach extract. First of all, the research background in this study was about integrating peach flavor in Kenaf leaves herbal tea to improve its taste and aroma. The stability of this product during 30 days of storage will also evaluated. Okay, we move on to the next part, which is the problem statement. Nowadays, consumers' preference towards herbal tea as a functional food are increasing due to health concern. However, based on the data by FFTC, the market size for herbal tea in Malaysia is still low compared to other countries such as Indonesia, Philippines, and Thailand. Therefore, producing flavored herbal tea may help in attracting more consumers from various groups of age, as the flavor can mask the bitterness of herbal tea. Okay, the first objective of this study was to formulate new flavor for Kenaf leaf herbal tea. Second was to study the stability during storage of peach flavored Kenaf herbal tea. For the hypothesis of this study, suggested that there is no significant interaction between the time and formulation of the stability of peach flavored Kenaf herbal teas, pH, moisture, and color. For this research, the null hypothesis was accepted. Next is the literature review, which consists of a few subtopics related with this study, which one of the subtopics is nutritional value and health benefit of Kenaf leaf, which based on the research journal, they concluded that Kenaf leaves contain a lot of phenolic content, which is beneficial for human health. Besides, consumers' preference towards peach flavor is keep on increasing due to its sweet and sour taste and attractive fruity aroma. Okay, this is the flowchart of this whole study, which started with the preparation of the sample, followed by stability test, data analyzing, and finished with a thesis writing. The next part is methodology. There were 3 parts and method, which started with the preparation of the sample. The raw ingredients were chopped into small parts and washed before weight. The ingredients were then dried in the dehydrator machine for a certain time. Then it will grinded and packed according to each formulation. For stability analysis, the samples were kept in 2 types of packaging, which were normal plastic packaging and vacuum packaging. 3 types of analysis were made, which were pH value, moisture content, and color of the tea. These tests were made on the first day, which the sample were still fresh, and on day 30. Next is the statistical analysis, where all of the data were analyzed by using Microsoft Excel to calculate the standard deviation. The data were also analyzed by two-way ANOVA and SPSS. The color analysis were made in Color Calculator website. Next is the result for stability test. For pH, there was significant difference between the pH of each formulation. This might be because of the ingredients difference in each formulation. And there were no significant difference between both packaging and no significance between formulation and packaging. However, there were huge difference of pH between day 1 and day 30. This might be due to the content of Kenaf leaf, which easily oxidize. For moisture content, no significant difference of all the factors. This shows that there were not so much moisture absorbed in each formulation. However, the least amount of moisture content was in F3, and the most of the least moisture content were on the formulation that have highest peach flavor. This shows that adding peach flavor powder might help in retaining moisture due to its ingredient. In the final analysis, the result for color showed that color of the samples on day 1 was lighter than the color on day 30. This might be because of the oxidation process throughout the time, which affects the color of the sample. The nearest color for positive control was F2 in vacuum packaging. In conclusion, the stability tests for moisture content were in good state. The pH value and color showed some differences between day 1 and day 30 of the samples compared to the positive control, which stays almost the same. Then, the nearest sample with the control were the F3 sample in most of the factors. As the recommendation, the study on more flavor for Kenaf herbal tea might help in getting the best formulation alongside with sensory evaluation analysis. Besides, study on the best type of packaging also might help in retain the quality of Kenaf herbal tea, as some studies stated that the light presence might influence the quality. Lastly, adding on natural preservatives such as ascorbic acid in Kenaf herbal tea might help in preserving the tea and make it more stable. In closing, during this project, I got opportunity to participating to competition, which I managed to get gold award for Agroric and second place for IVFIC. I'm so glad to be able to join these interesting events. That's all from me. Thank you for your attention.